This is the last part of the chapter focused on evolution. It's about cladistics, and I'd bet that you don't know what cladistics is. So this is the idea of showing the evolutionary relationships between organisms in the form of branching tree. It looks more or less like that. So what you are seeing here is a cladogram, the diagram trying to show how various groups of organisms separated during evolutionary processes to finally form the current state. And if it comes to the unit, a clade, it is any group of organisms which share the common ancestor together with this ancestor. So if you look at the diagram, you'll see that, for instance, you've got the clade which here starts with a blue arrow, and to this clade marked with this blue line, human, chimp, bonobo, and gorilla do belong. On the contrary to that, with the red arrow is the bigger type, is marked a bigger type of clade. And in addition to previous species, this clade contains also orangutan, gibbon, and old world monkeys. Of course, clades may be even smaller, like this one, in which we've got only chimp and bonobo. So the cladogram shows evolutionary relationships between organisms as a tree, and the clade is a group of organisms with their common ancestor. So human, chimp, and bonobo are the clade. Human, chimp, bonobo, and gorilla are the clade as well. But human, chimp, bonobo, and tarsiers only are not a clade, because such a formulation would exclude the rest of organisms which also share the common ancestor with them, like orangutans, gibbons, old and new world monkeys, and so on. So if we create a cladogram, what is crucial is after each branching, all groups belonging to the created clade should share some characteristics. And if you look at this cladogram showing relationships between vertebrates, you'll see that it is like that. So all vertebrates have vertebrae. These are these bony units in the spine. Then sharks are sort of left aside, while the rest of all vertebrates have also bony skeleton. This is their common feature, which all of them got after their common ancestor. Then all of them except of fishes have 4 limbs, and it includes amphibians, reptiles, birds, and mammals. If we go further, it will leave amphibians aside because the rest of vertebrates has so-called amniotic egg, the egg with special membranes which allow laying eggs on land. Among them, we've got 2 separate clades, one including hairy animals, so mammals, another one laying shelled eggs, and among them reptiles and birds, right? So we have to be sure that the clade we form shares some characteristics which is in common for all the members of this clade, because all of them inherited it after the common ancestor. If we draw such a tree, all branching points are called nodes. Such a node represents this moment in history when original one species splits into. And of course we are aware that actually such moments don't exist. Formation of new species is not something what happens in a moment. It takes ages in most cases. But on the diagram like that, we may show it in such a simplified form, and then the node signifies the common ancestor, after which there is splitting. What is important that this cladogram you can see here is not scaled. However, sometimes they are scaled. Then you can see the time axis at the bottom, and it will show how long were intervals between subsequent splitting events. So in such a case, not only can you assess which clades are younger and which are older, but when exactly the event of splitting took place. Now, if you look at this diagram here, you can try to understand which kind of questions IB may ask about it. For instance, look at the very top of the diagram. You can see there the duck-billed platypus, then American opossum, then marsupial mole and wombat, kangaroo, possum, koala, the set of marsupial animals. And if you look at the group which starts with American opossum, it is obvious that opossum, marsupial mole, and wombat are close relatives. But what you see here on this diagram means that actually marsupial mole is a closer relative to wombat than both of them are to American opossum. However, it is their next of kin. Clades may be of different sizes and different ranges. They can be really small, and sometimes they can be very large. It depends on how you want to define them. And now it is quite an important idea to get that in the ideal form, a cladogram would represent direct evolutionary relationships, and then taxonomy should reflect that. I mean, one clade should be given one common name in a biological taxonomy. But as you can see on this cladogram, at the current stage of development of biological taxonomy, this is not always the case, because what you get here is a cladogram showing relationships between reptiles, and look how it looks like. The oldest clade of reptiles, which separated the most time ago, are turtles. Then what sort of remained was split into, and the left branch formed lizards and snakes, which are close relatives to each other. But then the right branch evolved gradually into crocodiles, which are the oldest within this clade, dinosaurs, and birds. And now look, we call dinosaurs and crocodiles reptiles, but birds nowadays don't belong to reptiles. However, they share the common ancestor with all these creatures here. They are a bit separate. So we can treat them as a clade, but we have to be aware of the fact that nomenclature not always reflects the real relationships. How do we form cladogram? What is used as a background to decide about how close are relationships? As you probably know, the old approach had to be based on external features. So for example, here on the slide you can see feet of different animals, which can be used to compare similarities, differences, and so on. But there are many flaws in such a method, so IB assumes that the only valid way of preparing cladograms, if we assume that they should reflect real ancestry and evolutionary relationships, the only good source of such information is from molecular level. What does it mean? In most cases, cladograms are based on DNA sequences. However, you can also compare amino acid sequences in proteins. You don't do that like with naked hands, because it is impossible. Too many things to compare. In a moment I'll show you. Instead of that, you upload the data to some software, and the software will try to analyze all similarities and differences and then propose what is the most likely evolutionary tree showing these relationships. If you try to base such an analysis on one gene only, it can be misleading. So instead, usually many different genes are used, and the overall comparison is necessary to finally decide how to represent relationships between organisms. What is the background for this way of comparing organisms are, of course, molecular similarities between all organisms. So, all organisms on Earth use DNA as a storage of genetic information. Moreover, all of them use proteins as the crucial biological machinery. And finally, DNA encodes proteins using genetic code which is universal, the same for all organisms in the world. It allows a valid comparison between all organisms because all of them contain DNA, all of them contain proteins, and all of them are encoded in the same way. So you can compare DNA or proteins and see how many differences there are. Actually, it is based on that. Look here. Here we've got 4 different sequences of DNA. At first sight, they probably look more or less the same. If you take a closer look, you will start seeing differences between them. But if I ask you, which 2 of them are the most related to each other? Well, how to attack that? Are we comparing the first to the second, and the second to the third, and the third to the fourth? Oh, it's not enough. We have also to compare the first to the third, and the second to the fourth, and suddenly it occurs that it is not easy. So this is why we don't try to do it on our own, and we use special programs analyzing this kind of data. Look, if you compare the first and the second sequence, you will see 2 differences only. If you compare the first and the third, you will see 3 differences. Between the first and the fourth, again we've got 2 differences. And now what? Which of the sequences, the second or the fourth, is more similar to the first? You cannot say, because both are different with 2 bases. This is why, to make any valid comparison, you need much more than such a short piece of DNA. You have to analyze many genes, count many differences to make sure how close to each other they are. Here, not to torture you, the third and the fourth are the closest relatives, because actually they differ with only 1 base. This is the 6th position. In the third there is A, while in the fourth there is C. And the most distant are the first and the third. They have 3 differences. Comparing molecular sequences, we can also apply something what is called molecular clock. In other words, we can try to measure time of splitting between 2 species. How to do that? Do you remember how different variants of the gene are created? I hope yes, by mutation. So, originally there is 1 gene. Then suddenly it mutates, and we've got 2 different alleles of the same gene. They differ with 1 base. Then another mutation occurs, and there are 2 differences between these 2 genes. 2 alleles. The third mutation and 3 differences. The fourth mutations and 4 differences, and so on. If we assume that mutations appear randomly in general, then we can say that the rate of mutation in the single gene should be stable, constant, more or less. So the frequency of mutations is more or less constant for each gene. Why for each gene? Because genes differ in, well, how much they will be affected by the mutation. Look at the diagram. You can see that, for instance, for histone H4, do you remember what histones are? You don't? Yes, I know. Histones are proteins around which DNA is wrapped in eukaryotic organisms. So such a histone in all organisms is extremely the same. Like, differences in histone proteins are almost absent. And it is like they will mutate only with a rate like, I don't know, maybe 5 differences per more than billion years. So they mutate dreadfully slowly. Why? Of course, not because mutations hit them less frequently, but this is because if they are hit by the mutation, then they are destroyed so much that the organisms wouldn't exist, so it will not pass the mutation to the next generation, right? So such a mutation in a histone has almost no chances to be passed to the next generation. And due to that, the overall rate of mutation in histone is very slow. On the contrary to that, in fibrinopeptides, these are proteins responsible for blood clotting. In fibrinopeptides, the rate is very fast. It means that if mutation occurs in such a gene, then probably it will not destroy it, and due to that, the mutation will persist and will be passed to the next generation. And now, if we know the rate of mutation for the particular gene, like fibrinopeptide, or hemoglobin, or cytochrome C, and then we take some organism also possessing this gene, then looking at our organism and another organism, we can try to decide how long ago they split by the number of bases, which is different for them. Like, we take a cow and we take a horse, let's say, and we look at their gene of hemoglobin and compare them, and we say, oh, there are, I don't know, 20 differences between them. 20 differences means that 20 differences in hemoglobin would accumulate in about 100 million years. So it means that horse and cow split evolutionarily about 100 million years ago. Of course, I don't know if it was like that. But this is how molecular clock works. So, knowing the rate of mutation, you can try to assess how long ago the evolutionary splitting occurred and one species became 2 species. Now let's recall the idea of homologous structures. Do you remember them? We say that homologous structures are such structures which started as one, and then they gradually developed into different structures because they were affected by different selective pressures. And you should remember the example of pentadactyl limb, which has always the same template: 1 bone of arm, 2 bones of forearm, then the palm, then fingers. They can be differently shaped, different in number, but the general template is always the same, right? And looking at this set of structures, we can see both similarities, talking about their common ancestry, and differences, informing us about differences between selective pressures which shaped them like that. And this kind of sets of structures can be found in many cases. So another example you can see here are mouthpieces of different insects. even if they have totally different shapes, you can always find the same set of components in them. They are only differently shaped. So it is very similar to what we've got in a pentadactyl limb. Homologous structures are really helpful in understanding evolution because they can indicate common ancestry if we are not sure about that looking at other features. But much worse are analogous structures. These are structures which look similarly, but not because they had common ancestor, but just because they were affected by the same evolutionary pressure. And now look at the right set of organisms shown here. Bats have wings, and stoneflies have wings as well. But their wings are present and they look similar, not because there was some common ancestor of bat and stonefly after which they both get their wings. No, obviously not. What makes them having wings and these wings looking so similar is just the set of factors which affected them during their evolution. Selective pressure was the same, because if you want to use something to fly, it needs to have a special set of features, like it should be rather broad, rather thin, flat, you should be able to wave it, and so on, and this is why they look quite similarly. What's the problem, of course, is that such a set of analogous structures can be misleading if we try to deduce about common ancestry. Look at the fish and the dolphin. At first sight, it looks so similar that it is even hard not to think that they are directly related. You know that for a long time, people didn't know that dolphins are mammals. They look like fish, right? So, analogous structures may disturb our understanding of evolutionary relationships. Here, you've got once more a comparison of homologous and analogous structures. Analogous structures have no common ancestry, while homologous ones do. Analogous structures look similar because they were affected by the same selective pressure, while homologous structures were affected by different selective pressure, so they look different. The type of evolution shaping different structures into similar looks gradually is called convergent evolution, while the evolution which splits structures and from the one common template creates different things is called divergent evolution. The divergent evolution is informative if it comes to understanding evolutionary relationships, while convergent evolution may be misleading. Look here. Here we've got a evolutionary tree trying to show relationships between all groups of primates. This tree is based on comparison of 54 genes overall of the length of 35,000 of base pairs, so quite a lot of DNA. Numbers of differences were used to compare them and group them into smaller and bigger clades. Now try to find Homo. It is in this purple set in the middle of cladogram. Here we've got Homo, and you can see that within this cladogram the closest relative of Homo is genus Pan, while a bit older cousin is Gorilla, and so on. You can see that if you look at the simplified version, it is much easier to analyze direct relationships. The bigger is set of organisms, the harder is to say who is related to whom closer, farther, and so on. So in general, if we want to analyze like direct relationships, we look at smaller fragments of cladograms, or we simplify them just to look at bigger clades. And IB may give you a variety of tasks in order to check if you can read cladograms. Like, for instance, they can ask you, look at this cladogram and tell which animal is the closest relative of a squirrel. So of course you have to find a squirrel. It is here. And who's the closest relative of a squirrel? I hope you all can see that. Yes, this is guinea pig. Aktualnie po polsku jest to kawia domowa. Now, I'm asking, which animals are more closely related to humans than old world monkeys? Look, where are humans? Here. And where are old world monkeys? Here. So who is a closer relative of human than old world monkeys? All clades which evolved later from old world monkeys and their common ancestor. So it would include gibbons, orangutan, gorilla, bonobo, and chimpanzee, right? I hope you see that. Now, the third question. With which animals do tarsiers share a common ancestor? Like, tarsiers are here. And suddenly, I hope you should hesitate, because actually you'd see that there is no one good answer for that. Because tarsiers obviously have common ancestor with all these creatures here, like New World monkeys and so on and so on, and it was the first answer of most of my students. But then you look, okay, here are tarsiers. But for instance, if you look here, tarsiers, which are here, and bat, which is here, also had a common ancestor someday, much longer ago than with all these creatures mentioned previously, but still there was some common ancestor. Finally, we assume that we all had some common ancestor. So please be careful if the question is formulated in the way you can answer. Don't be sure and always check twice. Now look, elephant shrews, elephants, and manatee with dugong, they all belong to the same clade and they shared a common ancestor not so very long time ago, like here. Can you see them? Now, so which of them evolved first? Which of them separated the first and is the oldest of all these groups? Can you see that? It is elephant shrew. It split from the rest earlier, while elephant and manatee remained together for some time, and then they split not so long ago. So their group is younger than elephant shrew is. Do you see it? Such questions can be also asked, like which creatures are the closest ancestors, which of them split earlier, later, and so on. Cladograms may have different shapes, but there is one assumption which is actually broken in this cladogram, so it is not a good one. We always divide in two, like in dichotomous key. Not in 3, not in 4, always in 2. Then maybe the next division in next to, and so on, but in general we always think about splitting as about dichotomous event, right? And cladograms can represent like huge groups of creatures. Here we've got all metazoa, which is a really huge group of very different animals. While we can also produce a cladogram for a very small group of creatures, like here for dragonflies and different subgroups of dragonflies. So the idea of a cladogram of evolutionary tree may be used for showing the relationships for small and large scale, for small and long time distance. But the background will be the same for all of them. Doing that, so producing a cladogram, we try to reflect real evolutionary relationships between organisms.